introduction.
Sorry.
Yeah, so I'm going to talk about physiology, trying to understand vision, in this case, vision in a model organism in a mouse. So at the Allen Institute, we're different from a university in the sense that we are somewhere between a tech industry and a university. Our goals are basic science, in this case, to understand two species, mouse and human, at the cellular level. Everything we do works at the cellular level. So in humans, typically it's neurosurgical tissue, but I won't speak about that today. And then mouse, of course, it's behavior and physiology. And doing that in a large team. So our teams are between 20 on the low end and 300 on the high end. People working together, and everything we do is open science. So all of our data, all of our metadata, you can download it right now as I speak. It's all accessible. It's typically accessible 2 to 3 years before the final paper comes out, including all procedures, all reagents, everything like that. You can download, including Python notebooks for every figure in every paper. So this the paper here came out yesterday in Nature. So every figure you can reproduce from all the original data. It's roughly 100,000 neurons. You can download, and we provide all the Python code so you can reproduce every single figure and table, all in the service of high reproducibility. All right. So our understanding of brains remain very limited. If you look at neuroscience, where, for example, compared to molecular biology, some of us have already, and many of us hope to profit from the mRNA vaccines by Moderna and Pfizer. They're all due to an exquisite detailed understanding of the coding of how to reprogram, how to generate and reprogram particular short segments of mRNA to do our bidding. We're very far away from that goal in neuroscience. We know brains are assembled on the order of 1,000 different cell types. So they aren't just excitatory inhibitory neurons, as maybe your parents learned in college, but there are roughly 1,000 different types of neurons interacting. They are networked on the order of 10 to 50 to 100,000 other neurons, and this makes studying brains very different from studying social networks or any other networks outside biology. It's very difficult to understand this vast parallelization, this vast complexity. Neural networks, of course, they're very powerful, deep machine learning, etc., as you guys well know in Toronto, but they're very simple compared to brain networks. They typically have 2 or 3 different cell types and of course, primarily feed forward. While we understand, so I would say neuroscience is at the beginning, is at the end of the beginning of its first phase. If we compare it against the history of physics, we understand individual neurons reasonably well, but we have no idea how networks on the order of 10 to the 3 to 10 to the 11 components work. So although we have a wiring diagram of the worm C. elegans, the dirty secrets of neuroscience is there's no general purpose integrated model of its 302 neurons. We don't even have a good model, a model that reproduces for an arbitrary spatial temporal spectral input, the output of the retina. Although the retina is the simplest piece of neural tissue in our bodies. It has a known input, photons. It has a known output, action potential on 20 different types of ganglion cell axons, but there's no general purpose model. We don't understand the relationship between complexity of brains across species. Okay, that's a different point. All right, so we've focused over the last 10 years since I came from Caltech. We've focused of the 300 people, probably 200 people of these scientists and engineers and RAs were working on the mouse visual system, particularly the cortical system. And one of our big goals was to build brain observatories. So the idea is in astronomy, there are these very large observatories like the twin telescopes on Mount Keck or LIGO that are common facilities where everybody can apply to and after some selection process get access to observational time. This leads to highly reproducible science. Of course, in biomedical sciences, whether it's psychology, brain imaging, neuroscience, or cell biology, 2 out of 3 experiments cannot be reproduced for a variety of reasons. And that's definitely something that has to be addressed. So what we've built up over the last 10 years here in Seattle is these general purpose brain observatory that can record cellular level. So we are really talking here about the atoms of perception, which are neurons, which is not brain areas. It's not voxels that contain 100,000 neurons. It's actually individual neurons or groups of neurons in a very routine, reliable, and high throughput manner in behaving animals. So it's run by professional staff, so which has advantages and disadvantages compared to, let's say, highly motivated grad students or postdoc. It's different. We have several sets of instruments, particularly with multi-photon, multi-plane cellular fluorescent imaging that allows us to get at the genetic identity of the cells that we're observing. And we have these high density electrical recordings where we can record 1,000 neurons at a time in a single animal and then do that reliable, routinely day in, day out. So we get 100,000, 200,000 or more neurons. And it's open now to external scientists. So the advantage of working with a mouse is we have a very high resolution atlas available. We built this over the last 10 years, where the atlas is roughly 500 million voxels, but unlike voxels, unlike the 500 million voxels in the human brain, these are 10 by 10 by 10 micron voxels rather than 1 by 1 by 1 millimeter voxel. So it's roughly a million times smaller. And everything we do, of course, the other advantage of working with mice is they're isogenic, right? So they come from they have the same genetic background compared to humans that are, of course, very diverse, genetically speaking, much more diverse than just working with one particular type of mouse breed. So we use this atlas. So every time we do a recording, every time we do an electrical recording from a single neuron or from a group of cells, whether it's optical physiology, electrophysiology, or patches, we precisely place that neuron in its coordinate, in the 3-dimensional coordinate system inside the mouse that has this 10 by 10 by 10 micrometer pixels. We've built this high throughput pipeline. So by pipeline, I mean a set of processes that can be simple or very complex, where at the one end, let's say you have a mouse of a particular breed, a wild type mouse, or let me see, where's my cursor here? No, okay. Where at one end we put in mice, and then we do this whole series of using standard operating procedures by trained operators, where we implant the head pose, we do routine imaging, think of that like fMRI imaging, but it's done to locate in every mouse using whole brain imaging to locate the various brain areas. We train it, we do the recording. After the recording, we sacrifice the animal so we can track exactly, and we do 3-dimensional tomography of the brain, so we know exactly where we recorded from. Again, because location is critical to understand the brain. Even in V1, depending on what layer you are, and even if you know you're in layer 2, 3 in V1, depending exactly where you are in visual receptive field space is really absolutely essential. And so we have this entire, engineering team that sets these highly routine surgeries that has these gimbals, so we can return to the same place in the brain, 3, 4, 5, 6, 8, 10 times over several weeks, so we can track individual neurons using optical physiology. As you can see, it's really pretty cool. So these are different animals. These are different transgenic animals that express fluorescent tracers in different cell population. And we measure again and again their location here. You can see the area, primary visual cortex is that big blue area, and then the surrounding higher order areas. And then, as I said, we release everything in standard data set. So all the data today, you can just click on this link. You can just go to brain-map.org and you can download all the data. including all the standard operating procedures, and every single neuron, all the spikes, a few billion spikes in that, all of that can be downloaded in a standard data format, NWB, Neurodata Without Border 2.0, that's now widely adopted in the field. All right, so then you can do, for example, here we have a mouse running, so that's typically the mouse is running on a disk. It's head fixed, so we can do high resolution, in this case using optical microscopy. Here it's running past the movie, Touch of Evil. It's a nice movie because it has Orson Welles at the start of this movie. It's a fantastic movie, but it's also good because it has a single take, like 6 minutes long, without cuts. And here we can track we have cameras to track the eye, to track the body, etc. And here you can see this flickering activity is optical imaging in layer 2-3. So we can observe in real time, as it happens, individual neurons firing. And we all know this is the advantage of optical physiology. We know the genetic cell type of this. These are all excitatory layer 2-3 cells. It's sluggish. The time scale here is it's 30 hertz, matched to the fluorescent time scale. So that's a drawback. It's relatively slow, and it's in horizontal plane. But the advantage is you can come back to this exact field of view the next day, and the next day, and the next day, and you can identify the same neurons. So that's pretty cool. Now, the opposite, with the other technique we have, and there is no one technique that's best, as we all know in science, they're just different techniques that have different advantages and disadvantages. So this is a probe we helped develop, came out in Nature a couple of years ago, so-called neural pixel. It has totally revolutionized the field of electrophysiology, and most people are using this now, or moving towards using it. So it was built as part of an industrial consortium headed by Genelia, HHMI, Tim Harris. And it just uses more modern circuit technology, a CMOS circuit technology on pieces of silicon that are thinner than a human hair. So these are, the mouse version is 70 micrometer wide, 20 micrometer thin. So it's much thinner than a human hair, and 10 millimeter long. There are versions that have been built and that are tested right now as we speak for monkey, non-human primate. They're up to 45 millimeter long. And we are working with neurosurgeons to also get this into human tissue. So you have 968 channels along that shaft, spacing 20 microns, although there are now probes that we're testing that have spacing of 4 microns. So you get this incredible dense electrical view, which is why it's called neural pixel, very dense electrical view of a piece of tissue, in this case 10 millimeter, which gets us right through to the bottom of the brain, the hypothalamus. All the electronics, the amplification, the digitization, amplification, filtering, and multiplexing is all taken care at the base of this of the chip. So you only have 4 wires going out. So it's really fantastic, and they're fabricated at cost. Anybody can use them. You can order them neuralpixel.org. They're 1,200 euros, so really cheap compared to any other electrode around. So it's really revolutionized the field. There are optical versions now available where we're putting lasers and to do optogenetics. There are more advanced versions that are being manufactured, and there are versions that have electrical stimulations on board as well that are being planned now. So it's really changed the field. Noise flow is beautiful, 7 microvolts. Yeah. Very large data rates, very large data rates. So this has so far been tested only in mice, rats, monkeys, marmosets. Somebody asked me, there's no reason you can't use it in other animals. It weighs about 1/4 gram. But there are variants now that might even be smaller. And there are variants being developed that would be wireless. All right. So then you can so we build these workstations where the mouse is running on the wheel, as I showed you, with 6 neural pixels. So we have this pipeline up and running where we routinely every day record from animals running. and doing behavior that have 6 of these neural pixels implanted in here. We project the tracks of those electrodes onto a map of cortex. So again, because we do the tomography and the imaging afterwards, we know exactly where the electrode was, and so we can register each unit that we pick up. We can register, we know in what layer it is and where it is with respect to the other unit. So it really gives rise to very high precision electrophysiology and linking it ultimately to the brain, to the exact structure, which is essential. And so here you can see, so we have 6 electrodes recording, piercing sort of, as it were, here the 6 different visual areas, the primary visual cortex V1, and then the other, think of them as higher order cortex V2 and V3. We can also record simultaneously because, as I mentioned, these electrodes are 10 millimeter long, so they go through the entire brain to the superior colliculus. They go through the LGN and also LP, lateral posterior thalamic nucleus, which is really the mouse equivalent of the pulvinar. And so here on the right, you can see an example recording where we have, 3 different stimuli, different gratings moving at 3 different frequencies, and then recording from all these different areas, including hippocampus, because it's on the way between the cortex and the thalamus, and all the metadata that we track. So here, I try to make a visualization to emphasize for people who aren't so familiar, the difference between optical physiology and electrophysiology. So they're 2 different, they are by far the 2 most common physiological cellular level measures of neuronal activity in most animals now. And so the optical physiology gives you the genetic identity, so you know you're recording from a PV interneuron, or in this case from layer 2-3 excitatory pyramidal cells in a horizontal plane, your imaging plane. And typically there are several hundred neurons that you can see at 30 hertz, which of course is slow compared to cellular switching to action potential, which fire at high, action potential with is half a millisecond to 1 second. Compared to electrophysiology, so these are neurons, the baseline firing rates indicated by the diameter of that sphere. We can record cellular level activity along that track, that's the neural pixel track, from area AL, it's a higher order visual areas, from the hippocampus CA1, dental gyrus and CA3, and then from the LGN, the medial geniculate nucleus, and anything else at the bottom if we wanted to, like hypothalamus or midbrain. So that gives you very high resolution activity along a vertical track. It's at 30,000 hertz compared to 30 hertz, so it's 1,000 times higher, so that's much, much better. And of course, we have a much better fundamental understanding of the ontology, as it were, of action potential compared to the fluorescent signal. The drawback is we don't know the genetic identity of the cells we're recording from. We can typically say whether it's an excitatory cell or an inhibitory cell by the shape of its action potential, but we don't know that, one of these cells is a particular type of interneuron that only targets a different type of interneuron. That this technique is blind to, at least so far. So like any technique, there are trade-offs that you have to make. The beauty of it though, so this shows you one experiment with 6 neural pixels, where we can get the streaming data and you can analyze it. If you download it using our SDK, you can just look at it. You can see as the animal sees different visual stimuli, you see these pattern of action potential flowing over, and they're all recorded simultaneously. And so the data that I'm going to briefly highlight here is indicated on the bottom. So comes from this number of animals. And in each of those areas, LGN, V1, and then the higher order areas, we've recorded whatever, 1,200 units, 5,500 units. So you can really see that this just changes the entire field, right? Until maybe 5 years ago, 10 years ago, people recorded, a big study was 50 neurons, or let alone 100 neurons. Bill Newsome, became very famous on the basis of 202 neurons in MT. a smaller study was 30 neurons. So now we have 10,000 neurons, 50,000 neurons. And of course, you get a much, much better statistical and validity because, you just have so many more overwhelming number of neurons. Of course, it's still only a very small fraction of the number of neurons that the animal presumably has or that's involved in any one particular task the animal has to carry out. So the total number of neurons in the mouse brain is on the order of 70 million, 70, of which 14 million are in cortex. The comparable number in humans, the average number of human brains, at least in 4 aged Brazilian men, was 86 billion compared to 71 million, so a factor 1,000. And the number of cortical neurons in human is 16 billion compared to 14 million. So it's pretty much the biggest difference between mice and us is a factor 1,000. The 1,000 times bigger brain, roughly, in terms of volume, it's a liter compared to a cubic centimeter. Number of neurons is also 1,000 between human and mice. And so you can do all the beautiful physiology that people have done over the years in monkeys and cats and other creatures. You can do here, you can reproduce and you show the same stimuli to the same animal and you record from these neurons. So you do gabor, you do full field flashes, grating, stationary grating, drifting grating, start motion, natural scenes, and then movies, as I said. And you can record, and you can study each neuron, and all of that you can, as I said, it's in our SDK, so you can download and look at thousands of neurons, and you can see their firing rates as a function of temporal frequency and spatial frequency and all the gory data, most of which has never been seen by human eye. And so one thing in our experience, when we release these large data sets, there are many more papers that other people do from other labs that have no involvement with us that just analyze our data, just like in astronomy based on their surveys. I just like to draw your attention to the plot on the lower right. So that shows you we have an automatic way to plot the receptive field, the spatial receptive field, i.e. the area in visual space from which any one neuron can be excited by showing a gabor, a grating, or a movie or a scene. And so you can see the fraction of cells with receptive field is roughly around 70% and then goes down in the higher areas. And in hippocampus, it's basically 1% or so. So it's tiny. So that's a control. Because, of course, we wouldn't expect neurons in hippocampus by and large to have visual receptive fields. But units, the bottom line, units recording in all of our 8 areas. So we have 6 visual cortical areas, V1, LM, RL, AL, PM, and AM anterior. I mean, L stands for lateral and M stands for medial. And the 2 visual thalamic areas, the LGN and LP, i.e. the pulvinar, they all have 60% or more of the cells display significant spatial receptive fields. All right. So now we're trying to connect this database with a previous database that we published. We have a very detailed, it's our most popular product, a connectivity database. So we published this first in 2012, but then we keep on updating it. There are now several thousand mice, either wild type mice or transgenic animals, where we go and inject with a syringe, we inject a tracer, a fluorescent tracer, let's say, into one particular part of the brain, let's say one particular part of cortex or thalamus or hypothalamus. That's on the right. And then it's a fluorescent tracer, so it gets taken up by the neurons. Then the axons take it up and take it all the way to their synaptic terminals, to the end. And then we can measure this fluorescent signal. It's an enhanced GFP. We can measure it, and we can we sacrifice the animal after 2 weeks, and then we do this tomography where we slice and image the entire brain every 100 microns, and then we can reconstruct the density of the signal throughout the entire brain. And here we show you the logarithmic compressed dense sort of fluorescent signals. And so on the left hand you see is ipsilateral connections. On the right, you can see contralateral connections. And people have done deep analysis on this. So it's a brain-wide connectivity map now in several thousand mice. At the level, because we use specific transgenic animals, we can say, okay, let's only look at the connection of layer 6 or layer 5B extratelencephalic neurons. So neurons that have their cell bodies in the lower part of layer 5, but that project specifically outside cortex proper, let's say down to the striatum or to the spinal cord or something, and then look at the strength of that signal. So what we get images like this. These are these fluorescent images, and so this shows you 9 pairs of images where we have in a particular cortical area where we look at either neurons that originate in cortex and project to cortex, or neurons that originate in the thalamus and that project to cortex. And they have these very specific termination patterns where they either terminate only in layer 1 and in layer 4, or they have this very heavy layer 2, 3 and layer 5, 6 termination. And we used unsupervised learning methods to cluster, to do this unsupervised hierarchical clustering of cortical, cortical-thalamic and thalamocortical projections, where we classify neurons either into feed-forward or feedback connections. So this is sort of, if you think about the classical Harrefeld-Van Essen diagram in the monkey, we're doing something similar here, but in an unsupervised manner. We come up with these 9 different types of connections. And then we can arrange all of that in a hierarchy. This came out last year, or I guess 2 years ago now, where we can now arrange the entire, all roughly 40 cortical areas and with some 24 thalamic nuclei, we can arrange in a hierarchy. Okay, so on the bottom along the horizontal axis, we have something called the hierarchy score. So hierarchy score for one-step hierarchy, let's say the classical human visual, LGN to V1 simple cell, that would be a hierarchy of 1. Then continuing in that classical scheme from simple cells in V1 to complex cells in V1, according to Hubel and Wiesel, that's another step hierarchy. So from LGN to complex cell would be 2 steps in a hierarchy. And so if you look at Harrefeld-Van Essen, that hierarchy is like 7 steps or 8 steps, from retina to LGN to V1 all the way to hippocampus. This hierarchy here then is brain-wide, only is shallower. It is a mouse, it's much smaller. It's shallower, it only has 2, it only goes from -1 to +1, partly because it has massive sideways and parallel connections. If you actually look at the connection, it's a hierarchy, but the hierarchy with lots and lots of parallel pathways. And you can see that here where we split out on the right in these diagrams, either for the entire brain, where we can segment it into seven brain regions, just like human fMRI do for resting state activity, or we can look at specifically in the visual system. So now what we try to do, so we have 2 large databases now. We have a structural database of these patterns of anatomy of feed-forward and feedback connections in the mouse. And in different mice, but same type of mice, we have this database of functional database where we have spikes and where we can look at the relationship among spikes among neurons. So let's say we pick a neuron in LGN and we pick a neuron in V1. And we look at the cross correlation, and we see that, well, every time the LGN fires, the V1 cell fires 4 milliseconds later. And so we can infer something about the causal, that LGN might contribute in a causal manner to V1 with 4 millisecond delay. And how does this relate to the anatomical hierarchy? And so that's what we are doing here, where we are relating structure to function. And so if you look at panel B, so here we have two particular neurons. This is a neuron in V1 and a neuron in sort of in the next area, lateral medial, which you can think of it a little bit like V2. And in the example neuron in B, the LM neuron fired 3 milliseconds after the V1 neuron, consistently in a highly statistically significant way. So this presumption is V1, this neuron V1 projects to LM in a feed-forward way. And we do this here in panel C. We do this now for one animal for all the pairs between V1 and LGN and LM where we find a connectivity, that's 30. And then we average that across all the animals in which we have such V1 LM connections. And then we enter that on diagram E. So diagram E shows the mean cross correlation, the mean temporal delay in the cross correlogram as a function between the source and the target. D, the one to the left, shows you the anatomical hierarchy score between the difference essentially in A, between A, that there are various levels of hierarchy. So in D, we have the scatter diagram of the anatomical hierarchy score difference. E, we have sort of, if you want to think about the scatter diagram of the cross correlation difference, the time delay. And then in F, we can relate the anatomical hierarchy to the functional hierarchy, structure to function. We get this very nice, highly statistically significant relation that the bigger the anatomical defined hierarchy, the bigger the temporal delay, which makes sense if there is actually a hierarchy. So that shows the hierarchy that we see in the anatomy is faithfully reflected in the anatomy, in the functional connectivity, if you want, in the spikes. We can do the same thing in a model that produces this data very, very nicely. And because we have so many neurons, we can look at all sorts of other measures that people have looked at over the years, but in different animals, you know, in one or two areas, but we can do it in the same animals in all 8 areas. So for example, we can look at time to first spike in the top row, A, B, and C, right? So in A, you can see the average firing rates averaged over all animals for each of the areas. And you can clearly see the pink curve, that's LGN, peaks before the purple curve, that's in V1, that sort of peaks before the blue curve in LM. We can compute, because we have, I don't know, so, you know, 30,000 neurons here, we can compute the distribution of time to first spike in B, and then we can take the mean of those distribution. And again, they very nicely, anatomy relates very nicely to time to first spike. We can compute in the lower row, D, E, and F. We can do the same. We have an automatic way of computing the size of the receptor field. We can compute the distribution of sizes of the receptor field. And then we can compute the mean of those distributions. And again, as you go up the hierarchy, cells become, the average size of the receptor field clearly increases, as you would expect in a hierarchy. Here we do the same for a measure of simple versus complex. You know this from classical paper by Movshan and others in retina and in V1 in CAT. That if you have a simple cell that's really nicely driven by a sine wave as an LGN unit here, well, by the time you get to a higher area, the example here is area AM in the panel on the upper left, G, you have this much more tonic response. And so you can compute a modulation index, a log of modulation index. And again, you see a very nice hierarchical, in this case, inverse relationship that early on you tend to have things that are more simple-like, although they're never really cleanly simple and complex cells, right? The brain, if you look at large numbers, rarely behaves like you expect simple models do. And then while you go towards higher areas, you get more complex-like behaviors. And then you can also look at the time constant of response. So the way we compute that, not the time constant of individual neurons. So we compute those two measures. So what we do, we show whole field flashes. It's a very powerful, flash from gray to white, from gray to black, very powerful driver. We compute the autocorrelation of that, of those curves. You can of the responses of neurons on the left, and then we can compute the time scale of that. We fit an exponential through that. And then on the right, we plot the mean of those time scale of those. And what you see as you go up the hierarchy, the system responds slower and slower, a factor of 2. This is what you would expect. C.J. Wang has a very nice model that first made this point very nicely. So we see that here. This is different from the intrinsic time scale. When we do this very same operation for just when the mouse is looking at an empty screen. In other words, it's not driven by bottom-up stimulus, such as a whole field flash, just spontaneously firing. And we look at the autocorrelation and the time constant, and there's no correlation between anatomical hierarchy score and the spontaneous rate. So in other words, there's nothing intrinsically slower about neurons in the upper echelon of the hierarchy compared to the lower. But if you show a visual-driven input, earlier stages like LGN and V1 respond faster than higher stages. And then lastly, we also did behavior, and we're doing much more behavior now, but for this study that, as I said, came out yesterday, we also did behavior. So we have a mouse, it's trained to detect difference, change detection, right? So we flash up images, natural scenes, and occasionally the image changes, as you can see. And then if the mouse stops and licks, it gets rewarded. If it does so within a particular time window, it gets rewarded for properly detecting a change, an image change. It gets punished, quote, by a timeout if it incorrectly licks. All right. Mice can do this very well. Oh yeah, by the way, so here you can show, again, these are the visual areas we record from. Here you can nicely see the pattern of image flashes. Sometimes we leave out a flash. There's very interesting science to be done here. You can nicely see in hippocampus, by the way, the theta oscillations. I mean, that's one of the great things. If you have so much data, you can just look at it and see all these patterns. All right, so in the top row, you see what happens in C. This is like an odd man out task, right? Like people do in auditory deviant detection, right? P300, it's closely related to that. Same, different, except here it's images. So here in C, we plot in LGN and in V1 and in AM, what happens when an image changes from image A to image B. Then in D, we plot sort of the average, the population mean of that. And you can see when there's a change, as compared to pre-change, there's always a stronger response, which makes sense. The animal response, the neurons respond more strongly when there's a change in image compared when there's not. Now, and again, we can see this, this hierarchy. So the amplitude, so first of all, the time to first spike, also nicely in this behavior now, different mice, different behavior. Before there was no real behavior, now there's actually behavior. Again, it reproduces very precisely the previous relationship in very different animals. There's very nice relationship between time to first spike and hierarchy. Furthermore, we compute a change modulation index, right? The difference over the sum. So it's always positive, which means the neurons always respond more strongly to changed image. This is also true, but much less true when it's passive viewing. So it's not just some passive adaptation, right? So we have trials where the animal does, where we remove the spout, the water spout, so the animal can't lick. There's no reward, so it's just passive viewing, and we see some increase with anatomy, but it's much weaker than when the animal's engaged in a task. And furthermore, what's really nifty, so we can now decode, we can look at decoding and see how does so is it true that we can, for which area is it true that if we do a decoding, in this case using a random forest decoder, by looking just at, let's say, neurons, we randomly pick 20 neurons in, let's say, LGN and do a decoding, trial to trial, and you know, pick randomly 20 different neurons and all of that, and we can pretty much decode from all along the hierarchy. But then we say, well, how good is this decoding? What's its relationship trial to trial with the actual behavior of the animal? And then what you see, although LGN, so this shows you the problem of just doing decoding without knowing whether the animal actually uses that information. So for instance, from the LGN, you can decode whether or not the image changed with 68% probability on average. But if you look at how well does what your decoder gives you at the level of LGN with the behavior of the animal on that trial, that is essentially zero. It's a chance. Right? But it goes up. That measure goes up. And as you go up the hierarchy, it becomes like 0.8 if you once you record down in the midbrain close to the motor nucleus, which makes sense. So what this tells us that it's a hierarchy, and the hierarchy is also functionally relevant and sort of compatible with idea about predictive coding, right? That there's a change and the magnitude of that change signal becomes larger. All right, so that's essentially what I wanted to tell you. So we've built these observatories. Just like astronomical observatories, from the get-go, they're built with the idea of quality assurance and quality control. So quality assurance means you try to build very highly precisely engineered components that you keep track and measure by constantly, feedback information. Quality control means after you collect the data, you throw out data if it doesn't. So you have some constant operator independent quality control measures. So it's not that one day somebody comes in and he's more lenient, and the next day another operator, she's sort of more tolerant. No, we have some criteria or whole set of different criteria that we use for quality control. And if either the behavior or the psychophysics, the physiology doesn't fall within those criteria, we throw the data out, gets discarded. So typically 30, 40, 50% of the data gets discarded. To some people that might appear high, but of course, the problem is that most labs don't do that, or they have a very intuitive quality control. I don't think I'm going to use that animal, but that animal I don't want to repeat. And of course, the quality of the data speaks for itself afterwards. The work is done by trained full-time operators. That's what they do. That's what their job is. So they're unlike grad students that have to be jack of all trades, where you have to train and write code and do the experiment and evaluate and write the paper. Here you have different people. You have scientists conceive the experiments and write the paper, but you have engineers building it. You have software engineers doing the pipeline. You have operator doing the operations, etc. So it has advantages and it has disadvantages. All data, metadata, everything's accessible. The biggest trade-off is between standardization and flexibility. You build something very to a standard, you can really do highly reproducible things, but it's difficult to be flexible. If suddenly somebody discovered, hey, if I change this setting on this machine, I can do everything 30% better. Well, of course, in your lab, you would immediately do that. But we don't want to do that necessarily, because then suddenly we change something which is different from before. And if you do enough of those changes, you're going to suddenly compare apple to oranges. Yeah, so we've now opened this. We've done this three rounds. We've just got NIH funding to do this worldwide. So anybody on the entire planet, not just in the US, can apply to this, what we call open scope, where people can apply with their ideas for a project in mice, using behavior, using either optical multiplane imaging or neural pixels. If that proposal passes peer review in external double-blinded peer review, will promise to carry out the data and release the data and the metadata. Well, first we do some pilot to make sure we can actually do it. And then we commit to doing the experiments and releasing the data in a standard format. And you have to commit to analyzing it and publishing it. It's a pretty cool model. We did this over 3 years using Allen Institute funding, and now we're doing the same using NIH funding. All right, so I think I've gone over all these points. And with that, this is the team that does all of this this work. And of course, it's all due to the generosity of Paul Allen. And with that, I thank you for your attention, and I'm happy to take questions.
Oh, excellent. Thank you. Oh my God. Very impressive. Yeah, there's a bunch of little applause in the videos. Just so I can see who wants to have a question, there should be a hand up question that you can raise. That way I know. Otherwise, just unmute yourself and you can ask Christof your question. Are there hands up? I don't see any hands up, although I might be missing it. Oh, Jeff, go ahead.
Denise, sorry, Denise.
Yeah.
You need to stop recording.
Oh, yeah, let me stop recording.